Okay. So in this lab, yeah, we're starting off here, got to know the basics. So in this water worksheet, we've got the red oxygen, the two white hydrogens, and what's holding that single water molecule together for number 3, a polar covalent bond, one right there and one right there. Moving on. In the water molecule, shade the area where the electrons spend more time. Well, which atom is bigger, the oxygen or the hydrogens?
The oxygen.
Oxygen.
The oxygen's bigger. Excellent. That's a great point because that's going to attract more electrons. The protons in its nucleus of oxygen is going to attract more electrons. So the answer to this is more electrons will spend most of their time here on this side of the molecule, on the top in this picture, whereas they'll spend less time on the bottom. So what is the charge of electrons?
Negative.
Negative.
Negative, you got it. They're always negative. So where there is more electrons, that means will be more negative up here. And then less electrons down here would mean it's still negative, but it's less negative, which really is the same thing as saying a little bit partially positive.
Is it not? Are the electrons not shared where they meet the hydrogen? Is that not where they would be mostly hanging out?
Yeah, they're shared. They're buzzing around the hydrogen and then they're buzzing around the oxygen, but they're going to spend more time buzzing around the oxygen. Let's say 3/4 of their time, and then they'll come back to the hydrogen every now and then.
But I thought that the electrons, that was the bond that create, like that's how they bonded. So they're going to need to hang out where they're bonded.
No, the bond is already made whenever there's a sharing in general of electrons. That creates the bond because their valence shells are basically connected at that point.
And then they're free to roam away again?
Yeah, they're free to roam. Right,
Okay.
back and forth. You got it. And they just so happen, you know, since the oxygen has the new PlayStation, they happen to spend more of their time over there. Less time with the hydrogens have the old Atari 2600. If y'all even know what that is. Anybody know what that is? Nope. That's back in the 80s. That's when everything was like, you know, no graphics, just a bunch of blocks all over the screen. Yeah, so the electrons will spend less their time here. So we got less negative on this side, which we call partial positive. So you just should shade more electrons, more dots up here, less dots down there. That's all. Okay, so moving on to number 5. Oxygen will have a partial charge. Is it positive or negative?
Negative.
It's negative because it's more negative. We already said it's more negative. It's not a solid negative. It's just more negative. Whereas the hydrogens would be less negative, which means it would be known as positive.
You've already said this.
Say that again.
We should just.
Was that a question? Okay, I do want to remind those of you that may have your camera on to go ahead and turn it off because we're going to be recording this. Is everyone still seeing the screen?
Yeah.
Okay, yeah. Cool. All right. In your own words, what does it mean for water to be polar? We could either define it right off the PowerPoint or we could just explain casually what it means. We'll do both. Right off the PowerPoint, a polar molecule, water being one of those, of course, means that it's joined with polar covalent bonds. That's one way to describe it. A better way is to say there is unequal sharing of electrons across the molecule. Unequal is what I said, unequal sharing. And a casual way of explaining it is just to say that water has both negative and positive charges on each side of itself. So anything that has both charges, positive and negative, is polar. That's what makes water so reactive. It can react with anything with a charge. It stands next to something with a negative charge and it bonds to it on its positive side. It stands next to someone with a positive charge and it bonds to it with its negative side. So yep, anything with charges, other polar molecules or any ions that might have a charge. Okay, number 7. Whoops, one second. Take two water molecules, bring them close together. The hydrogen of one water molecule is attracted to what part of the other?
The more positive.
Oxygen.
Yeah, the hydrogen is actually positive though. The hydrogen of one water molecule is attracted to what other part of the water, what other water molecule? If the hydrogen side is positive.
Oxygen, because it's negative.
Yeah, so we're using the information in number 5, where we said hydrogen will have a positive charge in this case, or a more positive, partial positive. So since hydrogen side is positive, it'll be attracted to the oxygen of another water molecule. That's what I was trying to explain earlier. So for our purposes, just to make it easy, hydrogen is always going to be attracted to oxygen. Let's just put it that way, for the purposes of talking about water. And vice versa, of course. And that's how water sticks together, right? Those hydrogen bonding between hydrogen and oxygen of different water molecules. So here we go. It loads up the next question. This attraction between parts of two different polar molecules is called what kind of bonding?
Hydrogen bond.
There you go, hydrogen bonding. Perfect. The tendency for water molecules to cling together, is that cohesion or adhesion?
Cohesion.
Co is share, so we're sharing the same type of molecule. Cohesion is water molecules sticking to each other because of the hydrogen bonding, but it's called cohesion. Adhesion is when water sticks to anything else. Okay, a little experiment we do in lab takes just a second. If we just imagine this, in our minds, since we're so fancy now in our virtual world. Imagine a bead of water being placed on all 3 of these, right? Which one will soak in the best?
Paper towel.
Towel.
Right, the paper towel will soak in the best. Yep, because of the charges that paper towel has. So we'll just consider that all soaked in, right? For office paper, if I put that bead of water on office paper, what happens? It may kind of soak in. It may kind of soak in and be like a little flat, like the paper will bubble up because it doesn't fully soak it in as well as a paper towel. The paper towel has those electrostatic charges that match the water, so it blends in quite nicely. The office paper has a little more glue. It's a tighter fiber, so it doesn't mix in so well. Wax paper, what do we expect?
It's not gonna soak in.
Yeah, it just beads up there because the wax paper is hydrophobic. It's going to resist water. There we go. So why does water bead up on some substances and soak into others? Simply that polar molecules will blend with other polar molecules, but polar molecules will not blend with non-polar molecules. So that hydrophobic stuff you can spray on your shoes that prevents anything from staining or anything liquid, sticking to the shoe. There you go. Same old thing. We can skip the paperclip thing. That's just describing the surface tension thing. That's not that important. How about number 13? What does what you've learned about water explain why an insect can walk on water?
Is it because of the surface tension?
That's exactly 2-word answer right there. Best answer, surface tension. And that's due to the combined strength of hydrogen bonds on the surface of water. That's why you can float a paperclip on water. And in that example of 12, if you add soap to it, it just breaks the hydrogen bond and the paperclip falls. You can try it if you want. But yeah, the insect has, being a light, small insect, of course, can walk on water in that way because the bonding on the hydrogen bonding on the surface of water is still pretty tight to hold it. All right. So far, so good. Oh, let me get rid of these. My bad. Okay, here we go. In your own words, how would you define a non-polar molecule? And then give an example. So right from the PowerPoint, nonpolar means equal sharing of electrons. That's one way to define it. Another way to define it is that a molecule that has nonpolar covalent bonds. Or another way to define it is a molecule that exhibits no charge. Nonpolar, no charge. Polar is both charges, nonpolar is no charge. So it's like an all or none. And then who can give me an example of a nonpolar molecule?
Two hydrogens bonded together.
Yeah, a gas is a great example. Any gases are nonpolar, joined by nonpolar bonds. How about some lipids, right? And steroids, all joined with nonpolar bonds mostly. Lipids are very diverse. They can have a mixture of things, as we'll see, but they're going to be mostly nonpolar, like 90 to 95% nonpolar, or even more. So yeah, they would exhibit more in a hydrophobic or nonpolar environment. Here you go. So know your examples of each, polar and nonpolar. Once again, the example of a polar molecule was water. Some sugars and amino acids are also going to be polar. For nonpolar, that's going to be your lipids, steroids, and gases. You have to know those examples. And we're going to see them again as respective how they cross the cell membrane. So you'll be able to see them on the PowerPoint just the same. All right, this question is a little funny. I actually don't like it. I don't like that word interact because it's tricky. What they're really trying to ask you is, will polar and nonpolar molecules mix? And what do you think?
No.
No.
That's right. They don't mix, right? Polar and polar will mix together, yes. Nonpolar and nonpolar will mix together, yes. But polar versus nonpolar, nope, no mixture going on there. You can try to mix them, but they're going to separate again. So let's talk about this word interact. I doubt it will be worded that way on a test, but truthfully, everything interacts. Now, it does not mix, but I believe that the fact that it doesn't mix, that is in essence an interaction, isn't it? Just the same as if you walk into an elevator or walk into a room and you see someone that completely fits everything you've ever wanted, don't even talk to them and you just know you already want to like be with them, right? Some serious chemistry going on. That's an interaction. In that case, you're going to mix, hopefully, right? And certainly an attraction going on there. But what if you stand in a room or an elevator of someone you cannot stand, like a bad feeling, a creepy feeling, or you just don't like them at all, right? That's an interaction also, but no mixture. So you can see both scenarios are an interaction, but one mixes, one doesn't. Okay, so let's play with our sodium and chloride ions. Now in lab, again, we had these little magnets, these little atoms that are green and blue. So, for the purposes of just getting this right, I don't know if they're going to ask you the colors, to be honest, but just know that sodium is always positive, the cation. Chloride is always the anion, negative. So if something is labeled, right, and hopefully it'll label it as a chloride ion, they're going to want you to know how they interact together. So what happens when you bring these two ions together?
They mix.
Yeah, they attract each other. They sure do. They're attracted to one another because one's positive, one's negative. What if you bring two sodium ions together in 17?
Opposite of attract.
Yeah, it would, well, in this case, the word would be repel. Two sodiums would not stick together. They would repel each other. It's so cool in lab, you try to put these little magnets together and you cannot put them together. No matter how strong you are, they're not going to go together. I just love that kind of real-time chemistry. It will repel each other for sure. The forces are there. Two positive charges will not attract each other. They will repel. And just the same 18, two chloride ions will repel as well. Now, if we bring it close to water, remember our hydrogen and oxygen on the water. Which one was more negative, by the way, on the water?
Oxygen.
Oxygen, very, very good, because it carried more electrons around it. So what part, if the sodium ion comes close to the water, sodium is positive, which side of it will it attract to the water?
Oxygen.
The oxygen, right. So that's how water breaks up sodium chloride. That sodium will attach to the oxygen, and that water molecule will break the ionic bond that it had with chloride over here. So the sodium goes to oxygen. And what are the charges going on? Let's play the game here. And the chloride would go to the hydrogen, by the way, on the water, just to be clear. What is the charge for the sodium ion? Positive or negative?
Positive.
Positive, and chloride is negative. The cation is positive, the anion is negative. So which one's the cation?
Sodium.
Sodium, and anion would be chloride. Yeah, and if you missed that, chlorine is when it's in its natural state, non-reactive. When it's an ion, it's chloride. So there is a difference. And if that's on the test, you don't want to say a chlorine ion, you want to say a chloride ion. It's all in the details sometimes. So if we're being sodium chloride and put it in a cup of water, what's going to happen? What did we just say? Anyway, commonly, when you put salt in water, what happens? It dissolves, it breaks up, right?
Mix.
Yeah, you could say it mixes, right? It becomes solvent like that. Can you explain what's happening when you put salt in water? Do any bonds break? Oh yeah, those ionic bonds are breaking from it disassociating. Okay. So let's talk solutions. If you have a 15% sodium chloride solution, what's the solute? It is the sodium chloride. So that word solute, get used to hearing that. We're going to hear it a lot today, that's for sure, for our third lab. The solute is whatever you put in water, to be simple about it. In this case, they're talking about a 15% sodium chloride solution. So here's the question. If you see a beaker, a labeled beaker, right, of some kind of solution on your screen there for the tests, and it could be labeled sodium chloride, it could be labeled sucrose, it could be labeled anything, right? And it asks, what's the solute? You just named exactly what it's labeled. It's sodium chloride, right? Or sucrose, or whatever it might be. But if it says, what's the concentration of the solute, then you would look to the 15%, whatever number it is. So the question basically is the answer. If I tell you, here's a 30% sucrose solution, what's the solute? You tell me.
Gross.
In a 30% sucrose solution, what's the solute?
30%.
No, that would be the concentration.
The sucrose.
Sucrose, there you go. Now, if I said, what's the concentration, you would say 30%. Very good. I make that point not to make anyone uncomfortable, but that question alone is another one of those every semester questions. It's, I mean, what can I say? I've seen it every semester. So that, chances are you're going to see it again. And half the class gets it wrong because they answer it with the wrong answer. They say 30% instead of what the solute is, which is sucrose. And the concentration is 30%. So just making a point to get that straight. Trust me, I made all these mistakes in the past. I don't miss being on the student end of this. That's for sure. It's much nicer being the teacher. I feel sorry for you guys having to squeeze all this in your brain. I know it's a lot, but it'll be useful. What's the solvent? It's going to be water. So now we got another example here. Compare a 30% solution of sodium chloride. You tell me, which is higher, 30 or 15? The 30, right? So which solution has a higher amount of salt? The 30. It's no trick there. It is what it is. So which solution, 15 or 30, has a higher amount of water? That's a little tricky, isn't it?
The
15.
15.
If the 15, very, very good. Here's the math of it. If we know, by the way, when they label a solution, they're not going to tell you the concentration of water, because it's a given. They're going to tell you all these concentrations right here are always going to be the solute concentration. So if you hear, a 10% solution here, a 20% solution there, a 55% solution there, that's all solute. That's it. So whenever we see solute, we just have to automatically know what would equal 100%, because every compartment is 100%. So if we got 15% solute, it'd be 85% water. 30% solute would be how much water?
70.
70. So 85 over here of water is greater than 70 over there. So the answer to that one would be the 15% solution for the higher amount of water. And that's something we'll play with again today in third lab. So if you didn't like the way I got it here, we'll see it again later. Okay, great question here. I love this question. I would know this one. What's the difference between an ion and a polar molecule?
Molecule is two ions, like is two, like more than one or two.
You're close.
An ion
The
is just a positively or negative. It's just a particle with a charge.
Yeah, you got it pretty much, but let's clean it up a little bit. The ion is going to have an either or, positive or negative charge. It can't have both, of course. It can't. It's just an ion. But a polar molecule will have both charges. So polar molecules have both positive and negative on either side of itself as a whole molecule, whereas a single ion will only have or, positive or negative. Okay, let's play this game. What mixes well with water? Polar molecules, mix or no mix?
Mix.
Mix. Nonpolar?
Repel.
No mix. Yeah. Hydrophobic? No mix. Hydrophilic? Yes mix. Molecules with ionic bonds, or otherwise ions, you could say. Yes mix. Anything with a charge, guys and gals, anything with a charge will mix well with water. And you're hearing it right here, ionic bonds. That means you got some ions going on. It's going to mix well with water. How about lipids?
No.
No mix. Proteins. Now, you don't know much about proteins yet, but guess what? It's telling you mostly polar covalent bonds. So what do you think?
Yes.
Oh, yeah. Even the anions are negative. So there we go. Charges all over the place. It's definitely going to mix with water. Proteins love water. Let me tell you. That's what makes your body all healthy, keeping them proteins happy. What's it called when you make a protein unhappy? When it falls apart, it degrades. What's it called? We saw that in lecture.
Denaturation.
Denaturation. Excellent. All these bullet words like that, test questions all over the place. You got to know these terms. Okay, moving on to this application here. Let's see, talking about vitamins. Yeah, this is more of an AMP2 topic. Truthfully, I'm surprised they stuck this in here, but they're just wanting you to know that since fat-soluble vitamins are fat-soluble, means they don't dissolve in water. So how in the world can you expect a fat-soluble vitamin to move in the bloodstream if it's not supposed to exist in water? Well, what it does is it attaches to a protein, which is water-soluble, kind of like a raft. And then it makes its merry way through the bloodstream on this protein carrier. Lots of protein carriers in the bloodstream. But the hemoglobin is a protein. What does hemoglobin hold? Oxygen and CO2, right? Albumin is a protein. It holds lots of things traveling through the blood. So yeah, the simple answer to this is fat-soluble vitamins will have a transport or carrier protein that, that will enable it to travel through. Kind of like a little pod, you know? Okay, organic molecules. Here's another one of those. Do you know it or you don't? By this time, we've mentioned it so many times in lecture and in lab, actually. You should be able to name those 4 organic molecules. If you can't name them straight out of your head right now, well, you know what you need to work on. because that's where you need to be. I'm not saying you need to know everything, but certain things like your bonding, you need to be able to write out a note card right now. What are the 3 types of bonds? Ionic, covalent, hydrogen, and give examples of each, right? You need to be able to write out these 4 organic molecules and then also know what they break down to become at their smallest unit. These are certain things that you have to push yourself as the basics, to know for the tests, for sure. So let's just say we got proteins, carbs, lipids, nucleic acids. What do proteins break down to become in their smallest unit, which is called a monomer?
Amino acids.
Amino acids. How about carbs? Monosaccharides, right? Or simple sugars. And lipids, fatty acids and glycerol at a base level. And then nucleic acids break down to become
Nucleotides.
nucleotides. Excellent. So let's talk a little bit about proteins. These large molecules are polymers of proteins, of course. What are the monomers? We just said it. Amino acids. Which process would we use if we add 2 monomers or more together to make a larger molecule?
Dehydration synthesis.
There you go, our synthesis again. Making up bigger molecules is synthesizing something. And in the process, we give off water. Proteins are broken down into monomers by which process?
Hydrolysis.
Hydrolysis. Yeah, I love it. Have those straight. 4 structural levels of proteins. That's where that primary, secondary, tertiary, quaternary comes into play. This next one is an experiment, a brief experiment we do in lab. It's just kind of for fun. I'll explain it best I can like this. We have this tuber. It's basically just a blue, flexible tube, and we take a cup of colorful tacks. We throw those tacks on the table. Then we start randomly, the whole class just randomly places these colored tacks on this blue tube. And the blue tube bends and such, but right now it's still straight as you're adding the tacks. So let's just say out of all the colors of tacks, you see some red and blue tacks that you randomly put all across that tube. If you look across the room, you're probably never going to see 2 people that randomly placed the exact same colored tacks in the same sequence, right? That would be a miracle if someone did the exact sequence randomly. So this is exactly how we want you to think of amino acids coming together. The tacks would represent each amino acid, on this example here. So let's just say the red and blue tacks are attracted to each other. Well, yeah, we've got the tacks, of course, making those amino acids. Amino acids are joined with peptide bonds, as we know, right? But we're talking about a secondary attraction where each red tack will be attracted to a blue tack. So now think of a red tack being over here and a blue tack being way over there on the other end of the strand. Well, guess what? You need to touch that red tack to the blue tack, kind of like Twister. And you're going to be bending that blue tube to take that red tack and bringing it close to the blue tack, because for some reason they're attracted to each other, right? And then like 2 yellow tacks might be attracted to each other, or 2 clear tacks might be attracted to each other, that kind of thing. That's what the experiment explains. So all in all, you get this, roughly bended and twisted tuber that you end up with, and everybody's going to be different. So what that's telling you is that what makes the shape of a protein is this sequence of amino acids. In this case, the sequence of the tacks that we put in, the color sequence. So that's how the body builds proteins in all shapes and sizes. It adds different amino acids in a different sequence. And then with all this secondary bonding going on between the amino acids, not just the peptide bonds, but otherwise twisting and bending upon itself, folding, you could say. That's how we get these shapes. That's the long story short, or somewhat short. So let's do the questions. How many different amino acids are in the body? 20. What type of bond holds them together? Peptide. How many different proteins are in the body? Thousands. Definitely thousands. Too many to count. Okay, and number 6 just explains that, rarely, if all, if any, will the sequences be ever the same? So that would be no in pretty much all cases. Okay. So yeah, number 9 would be what determines how the protein is folded upon itself, and that would be the sequence of amino acids. The certain sequence. So guess what? Your DNA in the nucleus, it's got all the instructions for all these thousands of proteins. That's why the DNA is so important. It's basically the blueprint for your body. It knows exactly how to build this tissue, that tissue, everything to do with proteins. It's a big deal. So the level of organization in the tuber, most people are going to have a tertiary, where it's like a 3D. You know, it's twisted this way, that way, front, back, up, down, sideways, everything. That would be a tertiary.
Spell that.
What's that?
Can you spell that for me?
Oh yeah, tertiary. Tertiary.
Thank you.
Yeah. The 2 classes of proteins, we saw that in the PowerPoint. You guys can just dig from that to work on those, so we'll save some time there. Define denaturation. That just means when the bonds are broken and the protein becomes unraveled, and it's deactivated. The protein is deactivated. The structure is deformed, therefore the function is no longer. Getting into lipids real fast. Give some examples of steroids here, like testosterone, estrogen, cortisol, that kind of thing. Very important. Steroids have a huge impact on the body. They have free access, as we're going to see, across that cell membrane, since they're nonpolar. So since the cell membrane is nonpolar, guess what? Nonpolar molecules like lipids and steroids, they get to go right through the cell membrane, free access. So the same old thing here. Will steroids require, what will they require to travel through the bloodstream? Kind of like what we saw in that fat-soluble vitamin. They will need what's called a transport protein, that raft again that I was talking about. Something to bond to, in a way, a special bond, they can travel through the bloodstream. Like I said, the lipid family is very variable. It in some way can make small bonds with polar molecules. It's kind of crazy like that. It's not widespread, but every now and then that's what you get. So what are the building blocks and monomers of triglyceride? We did it before, the fatty acids and glycerol. Which process will combine these 2 together? The synthesis, right? Same old, same old. Will triglyceride mix with water? Nope, it will not. There's no charges to mix. Where do you find triglycerides in the body? All over. There's fat everywhere in the body, right? Certainly beneath the skin. Okay, let's move on to here. Insulation, of course, would be a good function and energy for the body. Let's look at this phospholipid right here. This is one type of lipid. It has a phosphate head and 2 nonpolar fatty acid tails. So let's label it. Where's the head? Right there.
The circle.
It's a phosphate. It's hydrophilic. That's what's crazy. Whereas these tails are going to be hydrophobic. Yep. And this is nonpolar. Nonpolar fatty acid tails, hydrophobic. Polar phosphate head, hydrophilic. So link those words together. Polar hydrophilic head, the phosphate. Nonpolar fatty acid tail, which is hydrophobic. So of the entire length of this molecule, would you say it's 90% nonpolar? Since the tails are nonpolar?
Yeah.
Oh yeah. So therefore it's going to behave in a nonpolar way. And that's what we mean when we say the cell membrane is more nonpolar. And there it is. There's your cell membrane. So you can easily see the thickness of this cell membrane. Check it out. Mostly nonpolar. A good 90% of it or more, right? Therefore, that cell membrane will act like a fence. it's not going to freely allow water-soluble molecules to pass through. It can't chemically, but it will allow fat-soluble or lipid-soluble compounds to pass through. And we'll talk about the specifics of that in the lab today. So yeah, don't worry about that. We'll get real specific on that transport stuff. Okay, guys, that is the water worksheet. That's your great study guide for the chem lab. So we're good 40 minutes in class now. We'll move on to the new stuff. And what I wanted to do, this lab today is all about osmosis. It is the osmosis experiment. So if you see that in lab, under the lab 3 module, yeah, this is the experiment we do in class. And I wanted to simulate a little bit of it today to show you what it means. So if you pulled up the lab 2 module and you're reading about this osmosis experiment and the beakers and such, I'm gonna put something on the screen to show you exactly what we're doing in lab, okay? Just give me a minute to get that going. All right, everybody seeing the whiteboard on the screen?
Yeah.
Great. So in this experiment that we normally do in lab, what we're doing is we're taking these dialysis bags, okay? It basically looks like a piece of plastic. It's very complicated, actually. In lab, it's so funny because I have all these students, you know, picking up these flat pieces of plastic. And then I tell them we're going to put 10 mils of fluid inside this piece of flat plastic. And they look at me like I'm kidding, right? And I'm like, no, it's magic. We're going to fill this with water. And they're like, well, how you get it open? Well, anyway, these are special bags that when you put them in water, they get all slippery and then they open. And then you realize that you can add fluid into them. So they have small microscopic holes. That's why they're called dialysis tubing here. Very, very, very small microscopic holes that allow water to pass through. Isn't that cool, right? Just like our cell membrane in a funny way, a different way. So we fill these bags, okay? Long story short, we fill these bags with either some kind of water or some kind of sucrose solution, right? And then we tie these bags up on both ends, kind of looking like a Jolly Rancher. So you're holding this little bag in your hand, about the size of a Jolly Rancher. It's tied on both ends like a candy wrapper. And I'm telling you that the water is going to come through that bag. Well, guess what? It's not going to leak into your hand. These holes are microscopic. You can't see them. The only way water is going to pass through is when it's driven on a gradient. So now we're going to talk about what a gradient is. A gradient is a pathway that defines how a solute, or water in this case, will move, which I'm talking about the direction, the pathway that water is going to move. So here we go. I'm going to draw some stuff on the screen here. We're going to have one beaker, for instance, and then another beaker. A total of 4, but I'm only going to draw 2 just to get the example across, okay? So let's just say we're going to put some water in that beaker, right? So here we go. There's some water there. There's some water there. And then we want to add a bag. So right here, I'm going to add a bag of water. Oh, actually, let me do that differently. My bad. Oh, I mean, I'm just going to do sucrose to make it better. It'd be a better example for us to do that. So this is going to be a cell, okay? Or a bag, in this case, in our lab. So we got 2 beakers with 2 bags in it. So now we're just going to add a certain something in the bag. So if I say we're going to add sucrose into the bag, then for our purposes of this experiment, red is going to be sucrose, okay? And blue is going to be water. So if I'm saying that I'm going to add a 10% solution of sucrose inside the bag, and now I say I'm going to add some, you know, the water is, of course, in the beaker itself. So we're going to say 100% water out here. Everybody got that so far?
Yes.
So all we did was add a bag of 10% sucrose, and the red is the bag, of course. The 10% is the sucrose and the 100% outside is the water. So now here's where we play with concentrations. First of all, what is the solute in the bag?
Sucrose.
Sucrose. Is there any solute outside the bag in the beaker?
No.
No, it's all water. But guess what? Since we have solute in the bag, we need to find out how much water is in that bag. If it's only 10% sucrose, how much water is in that bag?
90%.
There you go. 90%. Great. So here's another example. Before we get to talking about how things move, let me make it even more interesting. What if we say that there's 30% over here? 30% sucrose. How much water would be in the bag then? Inside the bag?
70.
Whoops. 70%. You got it. Okay, we'll just continue to add our 100% out here for water. There we go. So we got two scenarios, and these are the two scenarios that we actually see in lab that we're going to build in our normal experiment when we're in class. So here's the gist. They're very similar scenarios, but the difference is one bag has 10%, the other bag has 30%. So which bag has a higher percentage of solute?
The second bag.
The one on the
30%.
right. The one on the right. That means if we follow the rules for transport, that means we're going to have a steeper gradient on the right than on the left. And let me explain why. If you miss anything, don't miss this. Right? Here's the deal. Here's how to make things real simple, real fast. We can take osmosis and talk for hours, but I can explain it in one statement if you really want to know. If you want to know the easiest way to get this and always get it right, here it is. Water chases solute. That's what you need to know. Water always chases solute. So you tell me, where's the high solute concentration? In the bag or outside the bag?
In the bag.
Outside.
It's actually in the bag. The solute's in the bag, the solute, right? So which way is water going to go?
In the bag.
Water is going to chase that solute in the bag, right? What about the scenario on the left? Where's the higher solute?
On the right.
The higher solute's in the bag. So where's the water going to go?
In the bag.
In the bag.
In the bag. So what's the difference in these two scenarios? Well, they're both causing water to enter the bag, chasing the high solute, but the difference is that it'll happen faster on the right because it's a steeper gradient. So what are we talking about gradient? Let's get that straight, right? Here's the gradient. How much sucrose is outside the bag?
None.
0%. How about here?
None.
0%. Now we got gradients. Now what's the difference between 170 on the right?
30.
30. What's the difference between 190 on the left?
10.
10. So which is a stronger gradient? A steeper gradient? The 30.
The 30.
Think of a hill with a pitch of 30 degrees or 10 degrees, right? That's what we mean by gradient. So it'll just happen faster on the right. It'll still happen on the left and right equally. But if I gave this an hour timeline, guess what? It would happen faster on the right, right? And slower on the left. So we just indicate a steeper gradient. But all in all, that's how you figure out the concentrations of water. You just equal up 100% in each compartment. There's an extracellular compartment, which is outside the bag. There's an intracellular compartment, which is inside the bag. Each compartment has to equal 100%. Is everyone seeing that? How we did that? Let's do another example. How about this? How about, whoops, we'll throw something crazy out there. How about 35% over here, right? And then we'll throw how much sucrose we want to put in here in the beaker itself, 15% sucrose in the beaker. And then we'll make things really funny over here on the right. How about a 30% solution sucrose, right? And then only a 10% sucrose on the inside of the bag. Now things look all messy. Let's work it out. This is how you work through things. So now we got our solute concentrations already defined, right? So this is what they'll give you in a question. They'll always give you the solute concentration, like I said, and they want you to figure out how much water is there. So in each compartment, let's figure out the water. And might want to take a snapshot of this real fast before and after we do the water, so you can quiz yourself. So what will be the water in the 15% compartment on the left?
85.
85.
85% water. Remember, blue is water, red is sucrose. What's the water inside that cell on the left?
65.
65, right? What's the water on the right in the beaker?
70.
70, perfect. What's the water in the inside of that cell on the right?
90.
90. All right, this looks all messy. Let's make it easy. On the left, where is the high solute?
In the bag.
In the bag. So where's the water going to go?
In
In the bag, right? We can
Yes.
ignore everything else. We just want to know where the high solute is. We don't even have to draw the water. We just have to know where the high solute is, and that's where water is going to go. Water is driven to solute. Basically, solute is the Cheesecake Factory, and you are water, right? You're going to follow that cheesecake or whatever you like. Where is the high solute on the right?
In the beaker.
In the beaker, perfectly. So where's the water going to go?
Out of the bag.
Out of the bag. That's perfect. So here's how you'll be tested. Which bag will shrink?
One on
Because
the
it's
right.
losing water. The one on the right. Which bag will swell?
Left.
The one on the left. Perfect. So you're going to be asked that in a number of ways. You might ask, does water leave the cell or enter the cell? Does it shrink or swell? All of these kind of things, right? Or what's the net direction of water? Out or in? That's a third way they'll ask you. So what's the net direction of water on the right?
Out.
Out of the cell. Here's a twist. You guys, I love to carry you on my little raft here and then throw you off for a second. It's fun. You're going to hate me on this one. Does water move in both directions in both scenarios? In and out of the cell?
Yes.
Heck yes, it does. Water is sneaky. There will always be some water that wants to wiggle out like this, right? Or wiggle in like that. But the main drive, the main influence, the main gradient, the main pathway of water will obey the law. And the law is, here's the law of diffusion and osmosis. Water, being a passive transport system, water always travels from an area of high concentration to an area of low concentration of itself. So I told you the shortcut because now you guys all got it, which is beautiful. I told you water chased the high solute, and it does. Always, 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 Sunday through Sunday, every minute of the day, it never changes. Water will chase high solute concentration no matter what you're doing, trust me. But the other law, the real law that's presented in the book, is that it's going to travel down its own gradient. Okay, that's the verbiage you need to know. So what's the water gradient on the left? 85% on the out, 65% on the inside of the cell. Is water moving down its own gradient from 85 to 65?
Yes.
Yes, it is. On the right, is water moving down its own gradient from 90 to 70?
No.
Yes, it is. So we're still obeying the law, right? Water is traveling from an area of high concentration, which is 90, to an area of low concentration, which is 70. So that's the law, right? That's the law of diffusion, passive transport. But the shortcut is it's going to move toward that high solute. So learn which way works for you best. But I'm just letting you know, you're going to see so many crazy examples. Look for that high solute, and that's where that water is going to head to. But we just proved it by writing down the concentrations and proved the law that it travels down its own gradient from 90 to 70 and then from 85 to 65. Any questions on that? So this is the experiment we do in lab. You know, we let it sit for an hour and then we weigh the bags, and that's actually going to be what your quiz is. You're going to do this and you're going to simulate, you know, what the water loss was, I guess. Yeah, so yeah, bag on the left will gain water, therefore it'll weigh more. The bag on the right will lose water, therefore it'll weigh less at the end of the experiment. Perfect. All right, so now what we're going to do is just work on the actual data that you need to know for transport. So we're a good hour in at this point. We'll probably spend another 30 minutes going through the PowerPoint. It's all good. It is a lot to know, but I tell you what, this is the same stuff in lecture. And if you can get it today, like we just did, how many feel like they got it by the water chasing that high solute, by the way?
Already.
Perfect.
You
Man,
def-
that's the easy way.
you explained it very well.
Awesome. Well, yeah, you take a red blood cell, put it in an environment. That's what we're coming up to. That's another famous question. Shoot, I remember having that question in college and I couldn't stand it because my professors never made a bit of sense ever. Oh my God. I went to Ole Miss. It was a great school, lots of pretty girls, but oh my goodness, those professors are mean. Yikes. They do everything they can to embarrass you in class, and it was a different time back then. There wasn't all this, you know, political correctness. They would call you stupid right in class, you know. What are you doing here? You're so stupid. Leave the class. That's what they would say. Oh my goodness, a whole different time. I had one English professor, first day of class, say, you know, that he, you know, with a big smile on his face, that he welcomes everyone to bring a recorder to record the lectures so they can have big discussions afterwards, you know. So what does little Sliman do? He comes in with a tape recorder, you know, records the class, and he looks at me and says, "What are you doing?" I'm like, "Well, I'm recording the lecture." And he says, "How stupid are you? Do you have to record my words? You can't listen to me in class?" I thought I was in the Twilight Zone or something. So yeah, that was an easy drop. I immediately left this. Easy drop. Never look back. All right, so can everyone see the PowerPoint at this point?
Yes.
Excellent. This is showing us the difference between the two types of diffusion that we're going to see. Simple versus facilitated. Simple means that it's going to simply go right through the cell membrane. Check it out. Whereas facilitated means it's going to need a helper. That's what facilitate means. It means it's going to help. And that helper is this little protein carrier or channel. That's the help. It helps water-soluble molecules pass through. How about that? So water-soluble molecules need help. They need a facilitator. And this just happens to be a happy little protein. If you had to assign it a structural organization, would you say this is a fibrous protein or a globular? Does it look like a fiber or does it look like a glob? It's globular.
Gobby.
Yeah, this is a tertiary protein right here, right? Tertiary protein right there. Embedded in the cell membrane, its sole function is to help these little ions or molecules, whatever they might be, but they're polar, they're water soluble or something with a charge. Anything with a charge is going to need a little helper. Anything without a charge, non-polar, is going to go right on through because that's a non-polar membrane. That's the science that we're now putting in front of your face for you to know. All right, here we go. A little bit about fluids. Most of the fluid you've got in your body, 2/3 of the volume of fluid, is going to be inside the cell, intracellular. That's where it matters. That's where the metabolism's happening. And we have water outside the cell too, but it's just not as much. We call that extracellular fluid. These are what we call compartments. The intracellular compartment versus the extracellular compartment. We saw that just a moment ago with the beaker and the cell and the bag we were doing with. That was just to simulate, you know, a cell sitting in a solution, right? A different solution, a compartment. So 2 different compartments. And remember, each of those compartments has to equal up 100%. So whatever the solute percentage might be, we figure out what the water would be in that one compartment, right? By simple math equaling 100%. So a few types of extracellular fluid. Interstitial would be just the same regular fluid between all cells. It's called interstitial. But if we're talking about blood, we call that fluid plasma. We don't call it interstitial when we're talking about blood. If you take red blood cells and white blood cells, it's surrounded by plasma. So that's a unique word only for blood. So in other words, fluid on the outside of cells, if it's in the blood, is called plasma. But everywhere else in the body, it's called interstitial fluid. Here's your solute, any particle dissolved in a solution. The solvent will be what dissolves the solute, by the way. That's always going to be water in our case. Solution just being the mixture of the two. Okay, here's our rules. Here's the rules I was talking about as it's presented in the textbook. Passive versus active transport. You learn one, you automatically know the other. They're opposites of each other. So we call passive transport a movement requiring no energy. A movement of solutes requiring no energy, no ATP needed. So you tell me, does it take any energy to fall downhill? If you're at the top of a hill? No, it doesn't. You just
No.
fall right on down there, right? But it does take energy to climb up that hill, doesn't it? That requires energy. That would be active. So to go uphill is active transport, we're going to see. To go downhill, what they call it, is passive transport. And again, that has to do with the concentrations. Passive transport is going to move molecules or compounds or ions or water, like we just saw, from an area of high concentration to low concentration. Where do we see the high concentration of water on the left? What's the number?
85.
85, right? And 65 on the inside of the cell. So did water move down its own gradient, high to low? Yes, it most certainly did. Now here's the game. If we're talking about solute moving, which is not the experiment we're doing in lab today, by the way, only water moves through this plastic bag we're doing. But in real life, where is the higher concentration of solute on the left?
In the bag.
In the bag. So where is it lower? Outside the bag. So which way would sucrose move in this picture on the left?
Outside the bag.
There you go. It would go outside and water would come inside. That's how you get equilibrium, right? But in this experiment in lab, I want to caution you, our experiment for lab is only about osmosis. So only the water is moving. So in this case, the holes in the bag or the holes in the cell membrane are only small enough for water. No solute can come through. But in real life, I just wanted to throw that at you. The cell membrane would allow movement of both the solute and the water coming through. How about let's play on the right? Where is the higher concentration of water on the right? Inside the cell. Did it move down its gradient? Yes, it did. It moved down 90 to 70. That's downhill, didn't it? That's the gradient. Perfect. Hopefully y'all are getting that because it's same old thing in lecture. It really is. So we go uphill or against the stream, against the gradient for active, low to high, whereas passive is high to low. And this has to do with the concentration of anything, water or solute. So by the way, know those. Know these keywords, okay? No energy, downhill, link that together. High to low, link all those together. No energy, high to low, downhill. Write them down on a note card. Show off that you know it. Teach your cat or something. Like he'll care, you know? Okay, again, diffusion, movement of solutes, whereas osmosis is movement of water. Please know that distinction. Diffusion is movement of solutes in general, right? Osmosis is specifically the movement of water molecules only. So that's your definition of osmosis. It's still all passive transport. It's still high concentration, low concentration. We just got different forms of it. So here we go. If we move nonpolar solutes, right, like lipids, steroids, and gases, we'll do that on a function of simple diffusion. because they go directly through, simply through. Simply slippery lipids. Think about that. That's how I remember it all the time. Lipids, steroids, and gases, they just slip around, don't they? Facilitated. This is using polar molecules, sugars, amino acids, and ions, anything with a charge. Now, I'm not saying an ion is polar. Here's the polar molecules right there, sugar and amino. But ions do have a charge, so they also need help getting across. And that help comes in the way of what we call a facilitator, which is going to be that membrane protein, a channel or a carrier. And guess what? Osmosis is a type of facilitated diffusion, actually, because it also uses a certain channel to get through the cell membrane. So that's going to come up in more detail in just a bit. But yeah, definitely know the difference between simple and facilitated and those examples of the molecules that are passing. Whereas osmosis is only water, right? But it's all passive. The last one is filtration. This is just solute movement driven by hydrostatic pressure, like in the kidney. The blood pressure being pushed, you know, like that. So the pressure of water basically pushing something through, pushing solutes through, kind of like water resting on coffee, you know, pushing the coffee grounds, the coloring to come through. That'd be filtration. The pressure of the water pushing on the coffee in that example. Okay, so you tell me on this picture on the right, the beaker, where is the higher solute?
Outside of the.
Outside the cell, right? So that means that the lower solute's on the inside comparatively, as we can easily see. Where is there more water, on the inside or outside?
Outside.
Outside.
Inside. Believe it or not, as far as the concentration goes. I know you see water filled up to the top. Don't think about that. But the concentration of water.
Okay.
So if you just hated what I asked, that's great. Now let me put it to you in an easier way. Like we mentioned it first off, where's the higher solute? On the outside. So where's the water going to go?
It's going to chase the solute, so it's going to go out.
Yeah, that water is going to go toward that high solute, isn't it? Like that. So when you're presented with a picture like that and you're not sure which way to go, you look for that high solute, and that's where the water is going to go. That's the easiest way to do it. I'm telling you, no percentages needed. All we needed to know is where that high solute was. So in future terms, we're going to call this a hypertonic solution. Something that has more solute, tonic or tonicity, is hypertonic. And we're going to say that the cell sitting in it is hypotonic. Low solute, hypo, low solute, hyper, high solute. So you're going to be asked, this will be labeled A, this will be labeled B. What is A compared to B? Hypotonic. What is B compared to A? Hypertonic. Right? That's how test questions will come. And we'll play with that a little more. Simple things become difficult when you start, mixing around how they can be asked. I realize that. So here's our big difference between simple and facilitated, like we saw before. Molecules that use simple diffusion will go straight through, like nonpolar molecules. Molecules that need help will be our water-soluble molecules and ions. They'll need help in the form of a protein channel or carrier. Okay, good. Again, this is just a review of our types of passive transport. Please know those 4 types. Know which one carries nonpolar molecules versus polar molecules. And again, for osmosis, water chases solute. Those 3 words can save you so much trouble. It'll always be driven towards higher solute. That's the nature of water. It wants to equilibrate everything. But in doing so, realize it's always going to move down its own gradient. Definitely, definitely, definitely. Okay, so one more time on the principles of passive. This is what you need to know. Just another review slide. And there's your 4 types of passive transport. All right, getting specific about simple diffusion, nonpolar molecules. Here's the examples: lipids, steroids, and gases. Please know these examples of what's going to move through using simple diffusion. Remember, simply slippery lipids, if that helps. And gases, they're slippery sometimes too. I know that's gross, but hey, whatever makes you remember, right? We all have gas, don't we? I know, I'm still 14. Anyway, these are your 3 examples. Don't forget them. You're going to see them for nonpolar simple diffusion. There they're going right on through. Either in or out, doesn't matter. Do you see any proteins in this picture? Carriers or channels? Nope, they go right on through with no help. When we look at polar compounds, those using facilitated diffusions, they need help. And what is the help they get? Protein carriers and channels. Those are the facilitators. Solutes such as sugars, amino acids, these are going to be polar or slightly polar. Not as polar as water, but they're still polar. And then we get ions as an example. Anything with a charge will be using facilitated. And these are the types of facilitators we've got. We've got carrier proteins and channels. The difference between a carrier, this is more of a lecture topic, to be honest, not lab. A carrier is more specific on the shape, whereas a channel is just like an open door. As long as the solute can fit through the door, it'll go. But it has to be water soluble. The carrier is going to depend on a certain shape, right? So one carrier might be specific for a certain solute, is what I'm trying to say. And that's what we'll see in the example coming up. Here is your regular channel. Notice it says lipid insoluble. Anybody know what that means? Anybody from lecture?
Insoluble means that it doesn't work with water.
Well, in this case, it's lipid insoluble, which means the same as water soluble. That's a trick. It's a terrible trick, but it's a trick. So yeah, we got that in lecture. I made that distinction that lipid insoluble means water soluble. It's one and the same. So yeah, get that straight. It's just another tricky way of saying it. So this should say water soluble, but instead they chose to write lipid insoluble, which can throw a lot of people off. That just means that it's not soluble in lipids, which is what water is, you know, to be honest. So there you go. There's a channel. High concentration, low concentration, moving down the gradient. And sometimes they call it leakage, by the way. Gated is going to be something we cover in the neuron chapter and the muscle chapter when we start talking about when a cell is stimulated by a nerve, it'll stimulate some channels which are called gated channels. So we'll see that later. Right now, know the word leakage. I'll be mentioning that more. They leak down their gradient. And here's a carrier. Notice the shape of that hexose right there, like a sugar. Look at the shape of that carrier. It matches the hexose. So just understand that carriers are specific on shape. That keyword shape for carrier. So this is again facilitated, a sugar or an amino acid going through, whereas the previous picture would be more like an ion, to be honest, going through, like a sodium ion or potassium, that kind of thing, or calcium. Got lots of channels all over that cell membrane. The cell membrane is chock full of stuff. And here's our special channel for water. Look how cute it is. Look at these cute little water molecules. That's called an aquaporin. Please know that term.
They look like the shape of Mickey Mouse's head.
They do. They really do. Yep. So there's the oxygen with the 2 hydrogens. Yep. So this is just for water molecules. No other molecule is going to use the aquaporin. It's dedicated for water. And water is sneaky, isn't it? Water can slip a lot of places. It tries to slip through the cell membrane on its own, and it may succeed from time to time if there's little kinks in the fatty acid tails here. But 99.9% of the time, which is basically always, it's going to be using that channel. So you might see a question. Here's another one of those crazy examples. You might see an example saying, can water move through the cell membrane with simple diffusion? And the answer is yes, it does, but it's accidental, right? The main passage of water has to be through the aquaporin using osmosis. Okay, so that's not osmosis here with the dotted line. This is osmosis with the channel. And that's where water is always going to choose to go through. Every now and then this may happen accidentally, but it's basically negligible. But like, what can I say? In science, there is always these crazy, exceptions. All right, just 2 more slides here. One more thing to share with you. Another play on tonicity that we mentioned. And this is what's going to help you with your red blood cell example you've got in your homework for lab. So let's make it simple. Let's make it real simple. Notice how the solute concentration, we got 6 particles in each of these cells, right? But the environment that we're putting it in is different. Everybody see that? Good, good, good. We'll do more of this in lecture. We're just going to cover it real fast right now. Tonicity is an easy definition for it. The book may describe it differently, but this is the easiest way to describe it. Solute concentration within a solution. So think of tonic being solute, tonicity. The word hyper being increased, hypo decreased. Therefore, the word hypertonic means increased solute concentration. So which compartment has the increased solute concentration? Which extracellular compartment, right? This one. Here's the high solute, right? So let's play the game. If that's the high solute right here, which way is the water going to move in this scenario?
And
Out of it and towards it.
It's going to chase the high solute, isn't it? Always chase the higher solute. Always, 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 always. Comparatively, where's the high solute in this one?
And
Still the same healthy cell,
Blue.
but now in this environment, it's going to behave differently. So where is the high solute?
inside.
Inside. Perfect. So where's the water going to go? It's going to chase that high solute. Can you believe it? It loves to do that. Always, 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 always. That right there, snapshot that picture with your phone. This is so important. This will help you with so many test questions. It'll make hard questions easy. Water chases that high solute. Notice there's no percentages here. You don't need them. The question is telling you already. Notice the question is the answer. If you see a question, here we go. A cell is put into a hypertonic solution. Oh my God, there you go. That's the answer. Where's the high solute? In the solution that it just got put in, right? Hypertonic. Therefore, that water is going to leave that cell and chase that tonicity. If it's put into a hypotonic, it's just the flip, right? The water is, of course, going to come in the cell because comparatively, there's more solute in the cell in this case. But yeah, if you wanted to play the standard game, you would easily say that there's a higher water concentration inside the cell here and a lower water concentration out here, because guess what? Wherever there's high solute, there's low water. Wherever there's low solute, there's high water. But people don't like to hear all that. It gets too confusing unless you really think about it. So I just take the easier way of telling you that chase the high solute. That's where water's going to go. Now let's play the opposite game. In which direction would the solute move in that case? On the right.
And
With passive transport, the solute would move in. So don't let that throw you. Most questions, I will say, in our lab 3, and certainly even on this unit 1 exam, you're going to be asked which way the water is going to move. You're not, rarely will you be asked which way the solute's going to move. We're more interested in you guys learning this osmosis movement. So remember, it could be thrown at you a couple of different ways on the test. It could ask you which cell will shrink. Cell C or cell A over here? Which cell will shrink?
C.
So let's see.
This one will shrink, right? Because it's losing water. Which cell will swell?
A.
Over
left.
here on the left. That one will swell because it's gaining water. Which one will have a larger mass? A. Which one with less mass? Over here. Let us, if we're just going to assign that C. Which cell will lose water? This one over here. Which cell will gain water? So no matter how it's worded, you should get it straight. Which one will gain and lose water equally? Equilibrium. Isotonic. Because guess what? I'm not sure if I mentioned it. I talked so much today with 3 classes. There will be some water movement in that direction. A tiny bit, because water is sneaky. That's what we always say. But I'm presenting this for a reason, because it's going to be a term you need to know. Whoops. That would be a large one. A little bit of water out, but mostly in. So here's what I'm trying to say when I draw these small arrows. You're going to see this, okay? And don't be tricked by it. Will water move in both directions? Yes or no?
Yes.
Yes.
Yes. We don't want it to, but yes, it does. Water is sneaky. What can I say? It is sneaky. But here's the key. Here's the key word you need to know, and then we'll finish up with one more thing. What is the net flow of water? That's a word you're going to see. The net flow means the given or the larger amount of water, right? The net flow of water is out of the cell on the right. It's in the cell on the left, the net flow. So you're going to be asked that. Does water move in both directions? Yes, it does. What's the net flow? It's out of the cell on the right, in the cell on the left. What's the net flow for isotonic? There is no net. It's equal both ways. That's the beauty of isotonic. That's what we want, by the way. You never want to put a cell in the wrong environment because it'll die. It'll shrink or swell. This is what you want in real physiology. That's why it's important that saline is the right concentration in blood. Those red blood cells will shrink or swell if the saline is wrong, if the plasma is the wrong concentration. So yeah, isotonic is definitely what we want. Perfect. So you'll see that example and you'll relate it to your red blood cells in your homework. Great, 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 great. Last slide coming up. Talking about active transport here. What we want you to know real quick is that with active transport, of course, it requires energy in the form of ATP. That's what fuels this pump. Here's our sodium potassium pump right down here. Here's that ATP molecule right there swimming around. It's going to bond right there in that little nook that you can see. And once it bonds, one of those phosphates is going to be broken off, releasing energy, and that pump is going to move. And it's going to open up and put 3 sodiums outside the cell. It's going to push them out. And on the flip side, it's going to push 2 potassium ions in. So please know this data at the top. That's the nature of the sodium potassium pump. 3 sodiums out, 2 potassiums in. Please know that. Don't get them confused. I remember like 2 Ken, and that helps me remember it. 3 sodiums out, 2 potassiums in. There you go. And guess what? You're going to love this. We'll see this in lecture. As soon as sodium is being pushed out, guess what the gradient is? Where is there more sodium now?
Outside.
On the
On the
outside.
outside. Where is there less sodium?
Inside?
On the inside. Oh my goodness, we just created a gradient. So where is sodium naturally going to want to move on the passive transport side of things?
inside.
From high to low, right? So sodium will leak down passively, only so that it can be pushed out actively, and then leaked in passively, pushed out actively, leaked in passively, using a protein, of course, a channel. I just didn't draw the channel here, but that's what will be happening. So yeah, there's this happy dance. Sodium being pushed out actively, leaked in passively, pushed out actively, leaked in passively. Potassium, how does it leak? It's pushed in actively, and potassium will do what? It will leak out passively, right? The difference is there's a lot more potassium channels. So we'll get a lot of leakage of potassium leaking out, only so that it can be pushed back in again, only so that it can be leaked out again. You see the redundancy? Everything is cyclical. That movement of sodium and potassium constantly dancing back and forth across the cell membrane is what gives you cells that have life. That is the electrical buzz of life, the electrochemical gradient. It's very much there. That's what electricity is. That's what your nervous system is based on. And your consciousness, all in your brain. Same old thing on every cell. Every single cell will have this going on. It's quite amazing. And that's what we call a resting membrane potential, by the way. That's what we'll see in lecture if you already read ahead. Was that a question?
No,
Or just an amazement?
mind blown. Yes.
Oh, I hear you. It's enough to blow your brain. And what really sucks is that they don't really say it that way in the book. They they play with a bunch of big fancy words, but they never actually put it together. That is active and passive transport working together, but in completely opposite ways, right? Yeah. So here's what we get. The quality of active transport gives us these products. Please know this over here. The fact that sodium is being pushed out, like we saw up here, that's why I said, please know that, makes sodium the major extracellular cation. And of course, we know chloride likes to always hang out with sodium. So wherever sodium goes, chloride's going to want to follow. That's just the nature of the chemistry. Expect chloride to always follow sodium. And then if you got sodium chloride on one side of the membrane, where do you think water's going to go? Water's going to chase that sodium chloride because it's a solute. So there you go. We'll see that coming up in another lab. Talking about major intracellular cation, since potassium is being pushed in here, that's what makes it the major intracellular cation. And then anion would be proteins, because proteins are naturally being produced inside the cell. And they're so darn big, they don't leak out. They get trapped inside the cell, the proteins do, until they're told to leave through another process. So proteins are anions considered inside the cell, as well as phosphates, because there's always loose phosphates going around. So there you go. Are these 4 on the test? You bet. You will definitely see a couple of those on the test. And you will definitely see this on the test, the sodium potassium movement. Okay. Man, what a chock full lab, huh? Well, let me get rid of these little doodads. So the rest of the slides are really just some summary slides. I don't want to take up all your day. We've been an hour and a half. That's what I planned today. But this is just the details of active transport that we've already done, just to review. It's considered an antiport since the sodium potassium is going in 2 different directions, opposite directions. That's more of a lecture topic, though. No big deal. Honestly, as long as you know that these 3 qualities or principles of active transport, you should be good. And understand the quality, excuse me, well, let me get to where I was, so sorry. Right here, sodium out, potassium in, and then your major cations, anions. That's what you should run away with for active transport. Alrighty, I think that's it, guys. That's just another picture of the same old, and that does it. Any questions at all for today? Anybody out there?
Hey, sir, you said this class was be recorded, right?
Yeah, I am recording it. I sure am.
Okay, perfect.